Next speaker is Simon Ladouce, continuing with the old talk. Simon is a researcher in cognitive neuroscience at KU Leuven in Belgium, and with a clinical neuropsychology background. He investigates human cognition and behavior in everyday contexts and employing portable research methods to gather real-world data. His goal is to develop practical interventions benefiting individuals with cognitive or motor impairments and neurotypical populations alike, fostering broader well-being. I think you can mention the title of your talk yourself, so I will not, and thank you. Starting.
Thank you very much for the introduction. Hi, I'm Simon. I'm a postdoc in Leuven. And I would like first and foremost to thank the organizers for inviting me. It's really an honor to be here and presenting as part of such an inspiring lineup of speakers. So today I'm going to talk about the contextualization of neural and behavioral dynamics in everyday life through environmental sensing and machine learning. So the very title of this conference, Neuroscience of the Everyday World, really resonates with the philosophy behind my work and what is really driving my research efforts, because I'm a cognitive scientist and I've always been interested about knowing a little bit more about what is happening for us humans. How do we think, act, and perceive when we are actually doing everyday life activities. So thanks to mobile brain imaging methods and wearable sensors, we can now record that kind of meaningful signals during free whole body movements, and we can also step outside of the laboratory, so that's really great. That's thanks to the work of people like Daniel for the mobile EEG scene. But I want to be a bit of a Debbie Downer today and make the case that we can do more. And the next frontier is really to achieve the neuroscience of the everyday world. And I really believe that it's within reach, and that part of the equation is really to get a better understanding of what is going on in the environment and to map all this rich information that we get from all those sensors in relation to the context of where they are taking place. So I'm going to kind of illustrate all this by telling you my research story. So it all started once upon a time in rainy Scotland. The psychology department received or acquired a mobile EEG system, and I started my research journey. So I was really excited to do all that everyday life cognition research, but back then mobile EEG research was more looking like this. As you can see, EEG system suspended on top of people walking on a treadmill. So most of those early mobile EEG studies were actually validation studies to make sure that the signal was meaningful and interpretable. So a lot of those studies were using the P300 marker of attention. So it's essentially a neural signature of selective attention when target stimuli are presented. So the paradigms that we use to evoke those signals are very boring. So we present beep, 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 and then a bop. And 300 milliseconds after this bop, there is a P300 that appears, and you can do it also visually through a series of very boring stimuli presented on a monitor. So quite interestingly, those validation studies were all reporting that, yes, it was feasible to record those signals when people were walking. But they also noted that in comparison to being static, the P300 amplitude was reduced. So that became one of the main quests of my PhD, to actually try to explain what was going on there. So first study, we replicated kind of this finding, attenuation of P300 when people were walking versus staying still. Then we thought, okay, but what's special about walking? So there's two things. There is the physical act of walking, and then we are actually moving through space. So we kind of torn apart those factors in a factorial design. And interestingly, we saw that in healthy controls, it was not so much the physical act of walking that was leading to this reduction of P300 amplitude, but rather moving through space. So people being wheeled on the corridors. In a third experiment, we went a degree more in terms of abstraction, and we had lots of different conditions that I'm going to spare you the description. But essentially what we found was that it was both the inertial, the vestibular input, but also the optic flow that was actually leading to this reduction of P300. So this additional information that needed to be processed by the brain was actually taking cognitive resources away from the processing of all these beeps and boops that were presented. So that left me at the end of my doctoral studies with a bigger, more existential question, really. Are all those even-related potentials even real? Or are they like just a byproduct of these artificial experiments? So presenting series of sounds, they are not very ecologically valid, let's say. And this kind of rabbit hole took me a bit away from my initial goal of studying real-world, everyday life cognition. So I wanted to address the next question: can we capture neural markers of attention, such as the P300, directly in the real world during natural behavior? So for answering this, I devised the next experiment, which was a visual search task, a real-world visual search task. So I asked participants to come to the library and find books. We compared this real visual search task with a classic P300 visual, this time P300 elicitation paradigm. Okay, and you can see on the left side many trials that evoke non-target and target P300 on the left with the tablet condition, and then only one single trial of the visual search task. So the participants pick up a sheet of paper that give them the position of the book in the library. They have to walk there, they have to find the book, and then whenever they find the book, they return to the original position, and then they go on, and they had to find, I think, 40 books or so. So it took about an hour. So anyway, I think that the most maybe and not so talked about challenge of that naturalistic kind of experiment is really to find out what and when things happen in the real world. And that's my point with this talk. It's really, okay, how can we retrieve that information, and how powerful it can also be to interpret the results. So here we used, since it was visual events, we used eye tracking to, with a very kind of a priori naive approach, define those visual events. So whenever they looked for the first time at the target book, we basically said, okay, that's the moment they start processing, and that's where we are going to extract the EEG response from. And we did also the same for non-target books that were preceding that one, so we could compare the tablet condition with the real-world one. So a little bit of a parenthesis. Back then, I used a proprietary software of a very well-known eye-tracking brand to review those long recordings. And essentially what it means is that you want to annotate the data, and for every time they are going to look at one target stimuli or an object of interest, you are going to label them. And that is, you can trust me on this, it's definitely not an enjoyable or efficient approach. Highly time-consuming, prone to human errors, and definitely not scalable, and the replicability can be questioned. There's been other options based on the segmentation of the data into fixation and saccades. You can take snapshots of what is happening in the scene camera and then again annotate it yourself. But with 4,000 fixations per hour, you have a lot of work ahead of yourself. Another option that is not optimal, but maybe better for fixed setups, like this one, for example, is to put some QR codes or tags, and whenever there is a fixation that is falling within a range of those tags, they get automatically labeled, which can be nice. But in our case, we would have needed more than 200 of those, and it would have kind of ruined the fun of the experiment because they would have given away the position of the target books. So no visual search anymore. So fortunately, computer vision came to the rescue, and I think that it's a much more enjoyable process. So I'm going to come back to this a little bit more in the next slides. So anyway, back to the library data. We had to do a little bit more magic to retrieve those P300, but essentially by also using the tablet data, so the neural responses to the stimuli that were presented on the tablet, we could retrieve a subject-specific template. And with this template and the timing that was provided by this eye tracking data, we could retrieve the P300. So I got an answer to my question: yes, the P300 is real and can be found when we are looking for something in the real world, and those things are actual real objects that are embedded in the environment. Pretty much that. So also we want to go full on with the real world, but it also showcases the importance to have a degree of experimental control, and these what we call scalable experimental designs are very instrumental in achieving that, because without this template, we couldn't have retrieve the actual timing and align all those signals in the real world data. So moving on, I left Scotland, rainy Scotland, for sunny Toulouse. There I worked in a more applied field. We were developing BCI to support flight operations in an aerospatial engineering institute. I had the pleasure to try a lot of different hardware in terms of mobile neuroimaging, and I'm just going to give you my own personal experience, and my take-home message would be that for everyday life neuroimaging, what really matters is that we want systems that are easy to set up, kind of quick, comfortable to set up, because a lot of them are very not comfortable when your participants start complaining about 20 minutes in the recording. It's really bad. The quality of the data needs to be good over prolonged periods of time. Minimalistic systems. If you have environmental sensing to provide context to your data, it's even better. And the form factor needs to also kind of make sense, so people, are not going to be too self-conscious when they are using it. And I think that also it needs to be cost-effective. And if you want to move out in the real world, it needs to be robust enough, so if it rains or if it's windy, or it doesn't break so easily. Moving on, over the last year, last year I joined a neuropsychology lab in Leuven, so in Belgium. And in this lab, we study what happens after stroke. So we have people that suffer stroke, and a very prevalent symptom is what is called unilateral spatial neglect. So it's basically a failure of attending the side of space that is opposite to the lesion. And it's actually quite prevalent, but although there are existing assessment tools for it, they lack sensitivity. And often it's the case that people do not get any diagnosis or any indication. They go back home, they struggle a lot with driving, with finding their way around the house, and kind of interesting and striking clinical observation is that when you put a plate of food in front of them, they eat only half of the plate, because they can only process, or they can only attend to that side. And if you turn the plate, oh, well, there's more spaghetti. So anyway, it gave us a little bit of inspiration for the next study. We wanted to use my knowledge or my expertise with those wearables, and we wanted to kind of assess whether or not we could get a grasp of this problem when people were doing something that they would do anyway. So no more long testing and batteries that are not actually capturing what they are struggling with. We would just put eye tracking glasses on, and we would look at what they are looking at when they do something that they do every day, such as eating a meal. So the first step before moving into this clinical work, we wanted to kind of validate the approach. So we wanted to be sure that there was a spatial bias to record in the first place. And for that, we wanted to keep everything very ecologically valid. We used a real-world, a real-world distractor, which we all have, a mobile phone. So by placing a mobile phone in the environment, we basically manipulated, we induced a spatial bias. It's a very well-documented, effect in the literature. And so we had two conditions. 24 people came 3 times to the lab. We gave them a tasty plate of spaghetti, and they just switched their phone to airplane mode, and they placed it either on the left, right side, or not on the table. We put on the eye tracking, they ate the spaghetti, and then they came back for a 4th session where we presented a lot of different plates with or without mobile phones on the left and right, but this time on a tablet as a free viewing tablet task as a control condition. So that's the design of the tablet task. So we recorded a bunch of stuff. So eye tracking data gave us measures of all the different objects of the visual scenes were actually explored, but also gave us eccentricity measures, so how laterally the gaze distribution was throughout the course of the meal. And we also looked at head and trunk rotations throughout the course of the meal. This time I've learned my lesson, and I applied directly computer vision to extract visual features from the scene recording. It worked quite well. We trained a model to detect the objects of interest. There were not so many, and they were always kind of the same across sessions. So it was very effective. This is an example of what was happening in terms of the data. Oh, we've got the sun as well. So you can see actually that the metric, the gaze eccentricity metrics, was based on the center of the plate. So even though the person was moving their body and their head, we could use kind of landmark in the spatial environment to kind of compute the relative position of attention with respect to the environment. So anyway, let's go to the results. In the tablet condition, we replicated this spatial bias as expected. So by placing the phone on the left, we had a distribution of gaze toward the left side, and the same for the right side, and very central when there was nothing to explore on the sides. And much more interestingly, because the data was much longer and spread throughout the course of a meal, so for very hungry students, it took only 5 minutes, but for some others, it was 10 minutes. But we had this very robust kind of spatial bias that we could capture with just putting the eye tracker on. Interestingly as well, it was not just that the gaze was directed toward the phone, but also the object that was on the phone side was more explored. So a general kind of spatial bias was induced. We didn't find anything interesting in terms of head orientation and torso orientation, but these measures might be more interesting with stroke patients because we know that they tend to kind of lean toward one side, especially when they get tired. So anyway, the kind of take-home message is that yes, we can actually capture spatial attention during everyday life activities such as eating a meal, and we don't actually always need to introduce experimental probes in the environment but we can use everyday objects, and by extracting information about the state of the environment, we can get the contextual information to retrieve insight from it. So it kind of resonates with what Hugo has said yesterday. I think that part of the next frontier to really go into everyday life cognition is to get out of the lab and map cognition, the perception and action with what is going on in the environment. So I think that by tracking also the location of individuals as they move around real-world environment, and by mapping it with geographical information system, with the features of this built environment, for example, the space syntax that he mentioned, it will be really interesting to see how the architecture and the layout of cities can affect our cognitive abilities. I've been talking a lot about the visual modality to get information, contextual information about the environment, but I think that we can also retrieve meaningful information from other physiological sensors, so respiration and heart rate, so deviations from resting states or huge increase or decrease in variability can be informative of cognitive events happening, and so you can timestamp your other sensors with that. I think that also a timely aspect to look at is climatic features of the environment. So the temperature, the air quality are all features that might be interesting and that are enabled with integrating all those sensors together and contextualizing them. Right? I want to really make the case, so it's a bit of an extension of the graph, but essentially we really are in a position that allow us to really explore this interaction between the mind, the body, and the environment. And I want to showcase a new ongoing study that we are running, working in Leuven at the moment. So we are exploring gaze and gait dynamics during urban locomotion in healthy controls and in stroke patients. For this, we are recording eye movements, gait dynamics, and we are tracking the location of our participants. So this is essentially what the experiment looks like. So we've got gaze data, we've got the location, and we got also the gait dynamics. And you can see that we are applying computer vision to know what's going on. Yeah, Belgian people really like their bikes. All right, so this gives us very rich information, and I think that it's really interesting because we can look at the interplay between different modalities, and here the gait and gaze features can be integrated, so you can really look at the relation, for example, how fast someone is walking and how much they are looking at the floor, for example. In this idea of looking at spatial syntax, you can also retrieve information about the layout of the city. Here we are segmenting the data in terms of what type of terrain people were walking over, so on flat surface, on dirt paths, or on cobblestones that are a little bit more tricky to navigate. We can really compare those conditions in terms of gait and visual exploration. And also, actually, using, again, computer vision, you can get a sense of what was the environment at that time you were recording, because it's not something like the spatial layout that is a bit more stable. So how many people were there, because it has definitely an impact on visual exploration, for example. And that's really something that can be really powerful in interpreting, giving interpretation to the data. Yeah, I just want to show that, of course, the technology is really progressing rapidly, but it's still not perfect yet. So there's still need to be cautious and to review a little bit of it because, yeah, statues are not real people. So thank you to all the people, the amazing people that I've been working with, and thanks to you for your attention.
Thank you for this talk. We have time for 2 questions. I think there's one online, so okay, first come, first serve.
All right, so we do have a question from the Zoom. And so the question is, how do shifting spatial conditions, like moving from indoors to outdoors or from noisy to quiet areas, affect real-time neural dynamics captured through mobile EEG?
Well, that's a really good question. I think that there's been studies that were comparing indoor versus outdoor walking, but yeah, I haven't really done any kind of that direct comparison. I would say that in terms of data quality, actually, it's quite comparable. And thanks to the work of other researchers in the field, we really came up with strong and powerful ways to clean the signal, to limitate, the differences across those conditions.
I guess mine's quite a similar question. I come from a EEG and classroom kind of background, and the stuff that you're doing is really, really awesome. It's kind of to do with data quality, and if you experience any kind of, when you're comparing, particularly in the library study, for example, when you're getting people standing still doing stuff, how much data loss you have, how you preserve that, how do you have a different pre-processing pipeline, for example, when they're doing the kind of standard tasks compared to the kind of more naturalistic tasks. So there's really no guidance at the moment on how to, alter pre-processing pipelines. So your thoughts on that and how much data loss we can deem as acceptable when we move to naturalistic environments.
Thank you so much for the question, and it's actually the perfect opportunity for me to plug in something that I should have actually included in the talk. This is an ongoing discussion in the field, definitely. So everyone has their own recipe for dealing with noise, and there's been this EEG Many-Step initiative where we are trying to basically come up with guidelines and a best practice for the processing and the analysis of locomotion, EEG recordings during locomotion. And I think that, yeah, it's something that is in development at the moment, definitely.
Well, let me know when you find out, because that'd be really helpful. Thank you.
But the initiative EEG Many Steps is still looking for people to contribute data and to be on board with the data processing. So if you want to contribute to the project, feel free to reach out.
Awesome, thank you.
Okay, sorry, thank you so much for getting my question in. So, yeah, about the P300 study. So we've done comparable work, but in VR. And my question is, it seemed like when people were locating the target book, they were probably also scanning around to like adjacent books. So did you compare like for some kind of like amplitude difference between P300s to targets versus non-targets?
Yes, we were recording the brain responses to both when they were fixating on the target book for the first time, but also when they were fixating on the 4 preceding books. What we also noticed actually was that the preceding fixations were not located so close to the target books. So there was like kind of a peripheral processing that was going on already, and that was actually a struggle with the analysis later on, because we had this kind of misalignment in the neural responses. And when you average all together the single trials, then it doesn't work out. You don't get a waveform anymore. So we had to basically use that template matching approach to realign every trial, because there was some jitter, there was some temporal jitter across trials. Probably